Hello, biologists. Today we're going to talk about membranes, transporters, and sodium-potassium pumps. Here we have the cell membrane, and I want to make sure we note that it's more than just a fluid mosaic. A mosaic is a larger picture made out of lots of little pieces, which is why it is called a mosaic, because it's a larger structure, the membrane, but it's made out of lots of little components. And it's fluid because it always moves. Like these little components are not stationary. They float around, they change position, they bump into each other. The membrane itself is constantly butting into vesicles and uptaking things. It's constantly reworking itself. So it's fluid physically, but also in dynamic. And it's mosaic because it's made of lots of different components. I want to talk about the different components for a minute. The main component of our phospholipid bilayer is these wonderful little phospholipids. So here I have the hydrophilic heads. They are hydrophilic because they are water loving. Philic means loving, hydro water. So these water loving heads can deal with the aqueous solution outside the cell and with the aqueous solution inside the cell. So they can face water, they can deal with it. Their hydrophobic tails, they do not like water. They are water hating, they are water fearing. These hydrophobic tails are the lipid part of the phospholipid, and that creates a barrier so that nothing can like pop through easily. Only a few types of molecules can go right through this lipid layer, and we'll talk about those molecules in a minute. So this makes a really good barrier here. We also have cholesterol. Cholesterol is important for maintaining the fluidity of your cell membrane. Fluidity means like the viscosity of it. So is it loose? Is it tight? If you don't have enough cholesterol, your phospholipid bilayer can actually crack. And so we want to keep it nice and smooth. And that's what the cholesterol does for us. So here's another cholesterol. Now, if we have a protein that goes all the way through from the inside to the outside, we call it a transmembrane protein. Trans means beyond, so it's going through beyond your membrane, and that's a transmembrane protein. If it's stuck on one side, it's peripheral. If it's embedded, but not puncturing both ways through, it's integral. Integral proteins are often used to anchor the cytoskeleton. Can you see these light colored lines in here? That's showing you the cytoskeleton. So integral proteins could be used to attach to the cytoskeleton to give your cell structure. We're also going to have protein channels. So here's a channel. It's a channel because it's open. So things can just pop through there. It's like a door, right? You can close it. It's not always open, but if it's open, you can just walk straight through it. In our membrane, we're also going to have carbohydrates. So all these little carbohydrates here, those carbohydrates have lots of different uses. Sometimes they're for signaling, like it can help you identify self versus other. Sometimes it's for attachment. Sometimes it's important to your immune system. But those carbohydrates often display information. If you have a carbohydrate, you give it glyco. So that's sugar prefix. This is a sugar attached to a fat. So it's a glycolipid. Here's the sugar. There's the fat. Here you have a sugar attached to a protein. So it's a glycoprotein. Here's the protein. There's the sugar. This is a globular protein, which is describing its size and shape. And then that's pretty much everything in our list of membrane components that we need to know. All of these components, though, are moving and jiggling and floating. Like this carbohydrate here might move and bounce into this protein channel, or might move over here and bounce into that transmembrane protein. So everything is dynamic and changing positions. Think of it as having the same consistency as like a wax ball in a lava lamp. So it's very flowy. Our beautiful phospholipid bilayer is really important to keeping us alive. The big deal with it is because it's two layers of phospholipids, the tails orient toward each other, and that makes a really good barrier for keeping things out. So anything that's large, anything that has a strong charge, which we call polar, that cannot cross this barrier. Tiny little things that are hydrophobic, lipid-based things, those can go through this barrier because they do not get stopped by the phospholipid tail. An example is carbon dioxide can just diffuse through, oxygen can just diffuse through, steroids can often diffuse through because they're nice and lipid-based, so this lipid phospholipid barrier doesn't stop them. But most things that are large cannot go through. So it's a great barrier in the sense that it's semi-permeable. Some things can, most things can't. We also have really important proteins that are going to allow us to take things in and out of the cell. Now, if you are using diffusion, it does not cost ATP. And if you're diffusing right through the membrane, it's called simple diffusion. However, a lot of diffusion can't go straight through the membrane, but it still doesn't cost energy. It just goes through a protein. So a channel protein or transport protein. If it flows through a protein without energy, we call it facilitated diffusion. So facilitated diffusion is diffusion through a protein helper. The shape of your protein gives it different names. If it's always open, it's a channel. If it changes shape to allow certain things through, like this oval only accepts ovals, that would be a carrier protein. Many carrier proteins require energy to change their shape, but the idea is that the shape of the protein gives it a different type of name. One of our favorite types of channels is the aquaporin. Aqua being water, porin means it's a pore. So these are things that can just allow molecules through the membrane. Steroid hormones, they go through because they are lipid-based. This phospholipid tail section is not a problem for them, so they can diffuse right through. Carbon dioxide is small. It's not got a large enough charge to be an issue. Carbon dioxide can diffuse right through. Same for oxygen, super small. It will diffuse right through the membrane. And aquaporins, they are open. They allow water in all the time. So water is so important that there's a special channel. This does not require energy because our need for water is so great that we don't want to be paying for it all the time. So our little aquaporins allow water in and out of the cell constantly. Each one of these is an example of facilitated or of diffusion, one of them being facilitated diffusion. So when you think facilitated diffusion, think about these channels that allow things in, but do not cost energy. If a channel does require energy, or if a carrier protein does require energy, we get to talk about how we use ATP. So ATP is one of the most amazing molecules. ATP has a adenosine. That's this part right here, the pentagon shape. And then it has 3 phosphates. So 1, 2, 3. So that explains its name. Adenosine, that part, and then triphosphate, that part. Adenosine triphosphate is so important for your cell because that last bond between the second and the third phosphate, this one right here, it's unstable. That bond has so much energy in it. And because it's unstable, it pops and it exposes that energy, and that energy can be used to do work in your cell. So when you use that energy, we're releasing it. We pop that third phosphate off. So here it is over here. And we're left with adenosine diphosphate. So now there's only 2 phosphates. But the really amazing thing is this is reversible, right? So I can take the energy out, and then I can put the energy back in by reattaching this phosphate. So the conversion from adenosine diphosphate to adenosine triphosphate is continual. We're storing energy and then releasing it and storing it and releasing it. We have a special type of name for how that happens and what it looks like. So dehydration synthesis is when you are, taking your phosphates and you're adding them together and releasing water. So dehydration, you're releasing the water. And synthesis, because you're building something. What are you building? You're building 1 molecule out of 2 molecules. The reverse of that is hydrolysis or hydrolysis, depending how you want to say it. I like hydrolysis because it breaks it into two parts for my head. Hydro, water, and lysis, to break. So in hydrolysis, you're taking one molecule and you're splitting the bond between it, but we can't have these empty arms. So we have to take a hydroxyl group and a hydrogen. To get those, we break water. So we take one molecule, split it in half, and we split a water in the process so that each of these has a hydroxyl group on the ends. So that's hydrolysis, you're breaking water. You're breaking water to break a molecule. Dehydration synthesis, you're releasing a water to build a molecule. That's related to ATP use. So in ATP use, every time that we form an ATP, we're going to take two molecules and put them together. So that's us releasing a water, that's dehydration synthesis. Now we have this ATP, here's the adenosine, here's our 1, 2, 3 phosphates, and we have our high energy bond. We're going to use that energy to do something. In our case today, we're going to use that energy to make a protein change shape and allow something in or out of our cell. I think of it as like a little bit of electricity. So this ATP goes up to your carrier and it breaks next to it, and the energy zaps the protein and allows it to change shape. So you're going to break that third bond. It's going to release energy. And as it releases energy, now you have one molecule broken into two parts, and you need to break that water. So that's hydrolysis, so that each of those molecules is stable. And then you reverse it. You do dehydration synthesis, and then you do hydrolysis. You do dehydration synthesis, you do hydrolysis. What's the point? We're going to be using and releasing water to change molecules, and we have that high energy bond that we can use for active transport. When we talk about active transport, we're going to name them based on their shape, but also based on what they do. So a uniporter, it's going to take things in one direction and only one thing at once. So uni, think one. A symporter is going to take more than one thing in the same direction at once. And then an antiporter is going to take two things, but they're going to go in opposite directions. So sim is the same direction, anti is opposite directions. Here we have a visual of how we're using ATP to change the shape of a transport protein. I have my transport protein here. A solute has gone into its little pocket, and I want to bring that solute into my cell. To do that, I'm going to release the energy from that third bond of ATP, and that energy is going to allow a conformational shape change in my transporter protein. So now this solute can exit the protein and into the cell. Something to note, we know that diffusion and osmosis always go from high concentration to low concentration, but that's not true for active transport. We can take something and move it from low concentration to high concentration. We're basically crowding it. And we want to have concentration gradients in our body. Think about how you get oxygen and carbon dioxide in. We need concentration gradients to move molecules around, and this allows us to create one. Now, molecules don't like to be crowded, so you have to pay for it. If you're ever going against your concentration gradient, so from low to high, you're crowding, it's going to cost ATP. So active transport's the only one that can go against a concentration gradient. Endocytosis and exocytosis. We know that endocytosis is taking into the cell. We've got two types, one for liquids and one for solids. So test yourself. Can you come up with those vocab words? They both start with P. And then we have exocytosis, which is releasing. Both of these is a step-by-step process where you're going to take pieces of the plasma membrane. They're going to bud off, and they're going to create vesicles. So right here, this part of the plasma membrane used to be part of the exterior, and now it's part of the interior of the vesicle. The opposite is true in exocytosis. Here's your vesicle. This is the inside of the vesicle. It's going to fuse with the plasma membrane. And now what was the inside of the vesicle is now the exterior of the plasma membrane. This is a complex process. These little vesicles don't actually float around the cell. They're carried by motor proteins. Those motor proteins grab onto them, and then they have two little legs, and they walk along the cytoskeleton. They literally walk, grabbing the cytoskeleton, and they pull the vesicles behind them like little packs on their back. So they will carry them to where they need to go. And those motor proteins do cost energy. So exocytosis and endocytosis are active. They are not free. We also have a third type of taking in or out, and it's called receptor-mediated endocytosis. So here I have a visual of phagocytosis, which is solids, pinocytosis, and then receptor-mediated endocytosis. In receptor-mediated endocytosis, you only pull in the vesicle if you have the right receptor attached to the right ligand, whatever the ligand is. In this case, it's saying it's a metabolite or a hormone, but it's going to activate these receptors, and then your receptors will pull them into your cell. So it's really cool. It's a complex process. It's important to know that that does exist, but we're not going to get into the details of it. So endocytosis, solids, liquids, and receptor-mediated. I want you to take a moment and think to yourself, this beta cell is making insulin to be used in the bloodstream. What kind of transport is this? There's two answers. It's active, right? Because it's going to cost energy to move these little vesicles along. And it's also exocytosis. This would be a good time to remind yourself the steps of each of these processes and what happens at them. Practice question 2. Glucose is transported into cells at the same time as sodium. What kind of transporter is being shown? Glucose and sodium, two things going into the cell at the same time. And so this is a symporter. It even says it right there, symport transporter. Glucose is transported into the cells at the same time as sodium. This causes an increase of cellular sodium. High levels of sodium can lead to hypertension, which is an increase in blood pressure. Explain why using your osmosis vocab. If I have more sodium inside my cell is hypertonic in relation to my bloodstream. My blood would be hypotonic. Because I have more inside my cell, water is going to want to come into my cell. I'm going to retain water. I'm going to have more water in my bloodstream, and that's going to increase my blood pressure, hypertension. So if sodium is pumped into your cells every time we take in something else, how do we get the sodium out? And the answer is sodium-potassium pumps. So sodium-potassium pumps are extremely important to your body. So important that 20% of your overall cell's energy use can be used just to pump around sodium and potassium, which is a huge amount of energy for your cell on just one type of transporter. How does sodium-potassium pumps work? Here's a nice visual. We have one, cytoplasmic sodium binds to sodium-potassium pump. We're in the cytoplasm. We have all that sodium in our cell that we moved in that we don't really want. So we're going to pump it back out. Here's outside the cell. This is my phospholipid bilayer. Here's my carrier. So I'm going to add 3 sodium. I'm going to use an ATP. And as I use that ATP, this is going to change shape, and now the sodium is released. When the sodium is released, potassium will bind to the other side, 2 potassium for 3 sodium. When the potassium binds, it moves back to its resting shape, and now the potassium can enter the cell. So 20% of your cell's energy is used doing this, just this. If a cell must expend energy to pump sodium out of the cell, what is true about the concentration of sodium? We can only go against the concentration gradient using active transport. So it's got to be that the sodium concentration outside is higher. We are pumping it against the concentration gradient. A paramecium is a single-celled organism that are found in freshwater ponds. Paramecium are hypertonic to pond water. which direction is the water going to move with respect to the paramecium? If it's hypertonic, that means that there's more solute and less water inside the paramecium. Because of that, water is going to want to go from its high concentration to its low concentration. So water will flood into the paramecium. Water, less water, movement of the water. What would happen to a paramecium if this continued? I have a video for you posted on Blackboard for you to watch. In the video, you'll see the paramecium reacting to water flow. The way that it really does it is it creates these vacuoles filled with water, and then it expresses the water out. So it fills and pushes it out, and fills and pushes it out, and fills and pushes it out. It's just like a sump pump. But what if it couldn't work? What if the vacuole stopped collecting and ejecting water? Then we would see the cell would grow and grow and grow, and the paramucia would eventually explode and die. Sodium-potassium pumps are so important to us that I want to take a moment to talk about how they relate to neurons and how they relate to muscle cells. It's been estimated that you could use 30% of your calories per day in your brain alone if you are using your brain really hard. Which I always feel like makes sense because when I study really hard, I get really hungry. And so I always treat myself to a snack when I have a big study session. The reason that your brain can use so many of your calories is because it is reliant on sodium-potassium pumps for your neurons to work. So let's talk about how that works right now. Here is a neuron. We have two neurons in this picture. Here's another. Your neuron has a body. In the body, it has all the components of the cell that you've already studied. So there's the nucleus, there's the endoplasmic reticulum. There are tons of mitochondria because you need a lot of ATP for these neurons to work. The goal of a neuron is to take in information and transmit it to the next cell. The part of the neuron that takes in information is called the dendrites. So here are the dendrites. They are listening to other neurons and getting signals. And then if the signal needs to be passed on, it's going to send the signal down its long projection, which is called the axon, to the other cells that it talks to. The place where it talks to the other cells is called the synapse. So these synapses allow this cell here to talk to this cell. Neurons work with electrical impulses. So it will take in an electrical impulse and it will send on the electrical impulse. Each electrical impulse is about the same amount of electricity. What changes is the frequency of sending it per unit of time. So it's not like more or less electricity, it's just one electrical impulse. The electrical impulse has to be moved through the membrane of the axon all the way down, and that's achieved with sodium-potassium pumps. So let's look at this a little bit closer. Here, I have outside my neuron and I have inside my neuron. When we send a signal down the neuron's axon, we call it an action potential. An action potential is literally just a temporary change in voltage. Voltage is the measure of positive and negative in electricity. The membrane potential is the potential for a change to occur in the membrane. And that happens because of these sodium-potassium pumps. So let's check out this figure here. I have my sodium-potassium pump. Sodium is going to be round. Potassium is going to be a diamond. I have a channel protein. This one's for potassium, and this one is for sodium. So this is outside my cell. This is my axon membrane with all the proteins embedded in it, and this is inside the cell. What do you notice about the different ions? Way more sodium outside the cell than there is inside the cell. Okay, so let's talk about how these membranes do an electrical impulse. The first thing that's going to happen is your membrane is going to be sitting. It's got its resting potential. That's about at negative 70 millivolts. And we can measure this. We can literally stick little electrodes into neurons. So negative 70 millivolts is just chilling. Then it will get the signal and it will make a temporary change in the potential charge difference between the two sides of the cell by going more positive. This is called depolarization because polar means that there's two opposites apart from each other. In this case, it was a positive on one side and a negative on the other. Depolarization means that those are going closer together. So they are no longer polar, they're depolarizing. After you have this spike in charge, your cell is going to try and reset itself. That's repolarization. It always goes a little too far. That's your undershoot. And then it goes right back to resting. What does this have to do with membrane channels? Let's look. So here I have my scale again, or my graph. I have resting state. I have my depolarization. I have my repolarization, the undershoot, and then back to resting state. Let's look at what's happening with the channels. So here is my inside of the cell. Here is my plasma membrane, that yellow bar, and then outside of my cell. When your cell, your neuron, is resting, it's not sending a signal. These sodium channels are closed. The potassium channels are closed. Now, when my cell gets a signal, and that signal can be any kind of stimulus, it could be being touched, it could be a temperature change, it could be a thought, maybe I have a feeling, because this is all in my brain where all my thoughts and feelings are. Something happens, and my cell is going to create an action potential. It's going to send one message from one neuron to another. That is the depolarization event, and that is caused because the sodium channel opens and sodium floods into the cell. Look at the charge of sodium. Sodium is positive. In general, the inside of the cell is more positive than the outside of the cell, which is more negative. As sodium floods into the cell, the charge across my membrane starts to get less and less different. I'm putting more positive in, I'm depolarizing. Sodium's going to flood in until those sodium channels snap closed at about 30 millivolts. When they snap closed, sodium can no longer go in, but note that the charge inside and outside of the cell has flipped. So where it used to be more negative outside is now more negative inside. And my cell does not like that. It wants to put it back. So instead of just pushing those sodium back out, I cut off the flow of sodium, I open the potassium channel, and now potassium floods out of my cell. That positive charge is going to leave the cell. That allows it to repolarize. All of the positive charges go back out, and it retains the more positive outside than more negative inside. When that is done, it goes a little bit too far. It pushes too much potassium out. It shuts that gate. And now I need to redistribute the sodium and potassium back to the resting state. So right here, to redistribute it, I'm using that sodium and potassium pump. Right here, sodium and potassium pump to put everybody back where it should have been. This is so cool because those pumps, those channels, allow an electrical impulse to go from one neuron to another neuron. What's really amazing is most neurons don't touch. At their synapse, there's a little gap between them. And to get the information from this axon to this dendrite, I need to transmit a signal across that gap. So the way that your cells do it is an action potential will come down this axon. That's that flip of charge created by the sodium and potassium channels and then reestablished by the sodium and potassium pump. That flip in charge comes all the way down, and when the axon terminal, which is the swollen part right here, sees that there's been an action potential, it will take vesicles and it will do exocytosis, and those vesicles will release a chemical into the gap between your two neurons, which is called the synaptic cleft. In this space, those messengers will then go to the body of the dendrite right here, and it will receive it as a signal. So the action potential causes the release of vesicles using exocytosis, and these little molecules send that signal on to the next cell. These molecules, because they're being released by a neuron, are called neurotransmitters. One of the most common types of neurotransmitters in the body is acetylcholine. So here we have another visual of this. We have the axon of another neuron right here. It's sending a signal down. We're looking inside of the axon terminal. The signal is being sent down. It's causing these vesicles to release their neurotransmitters, and the neurotransmitters are binding to the receptors of the cell below it. So this is sending a signal from one neuron to another neuron. And as those neurotransmitters bind, they often create another action potential in the cell below it. So here you can see a swap where the positive and the negative are switching place. That's our action potential. So we have channels and a protein core, and we have exocytosis, and you are using that every time you think or feel or memorize or are aware of your surroundings. Anything you do in your brain is using all of those components continuously. And it's just so amazing. And not only are you doing it in your brain when you're thinking and memorizing and feeling, you're also using it for all of your muscle movements. So we're going to have neurons that control muscles, and that's called a neuromuscular junction. Here's a neuromuscular junction. This neuron has dendrites. Those dendrites are getting information. The information is sent down the axon in the form of an action potential using the sodium and potassium channels and the sodium and potassium pump by changing that electrical difference by depolarizing and repolarizing. That signal gets to a muscle. There's a synapse between the neuron and the muscle. Exocytosis releases neurotransmitters. Those neurotransmitters tell your muscle to move. This is so cool because I'm doing this movement right now with my hands. I am firing trillions of neurons and trillions of muscle fibers, not trillions of muscle fibers, I don't have that many, but just a ton of different molecules moving around my body because my body is telling my muscles to move. So it's just an incredible process, and so much is happening so quickly for it to occur. Now, I think muscles are really amazing. All your muscle movements are moved by contraction. Contraction means tightening or shortening. So if you do like a bicep curl like this, my bicep here is shortening, and that's pulling on the bone of my forearm, and that's what's making my arm move. To move it back, I'm doing the opposite motion, so with my tricep. But every movement of my body is a muscle squeezing, just shortening. They can't extend, they can only squeeze. So for any movement I have, I'm going to have an opposite movement in the opposite direction with a different muscle that is shortening. I've got a quick visual of this because it's so fascinating. So here we have an axon. It's coming down. Here's the synapse. This synapse is interacting with a muscle fiber. We can see the little junction between them. Let's zoom in. Here's my axon. It's getting the signal that it wants to send a neurotransmitter to the muscle cell so that the muscle moves. These orange Cheeto-looking things, those are all mitochondria because this takes a lot of energy. So we have a ton of mitochondria involved. In our axon, we are sending the action potential. That's going to cause these vesicles to fuse to the plasma membrane doing exocytosis, and they're going to release their neurotransmitter to the muscle cell, who's going to receive the signal and will respond. The neurotransmitter that we use for muscle cells is called acetylcholine. So acetylcholine, that's this word right here. Acetylcholine is going to bind to the receptors on the muscle cell, and that's going to tell the muscle cell, hey, you need to shorten. Here's the zoom in. I have my vesicles. There's acetylcholine. This is the muscle cell. There are the receptors on the muscle cell. The receptors will bind to the signal, And that's going to allow sodium to flood in through a sodium channel. So we're using even more channels. That change in sodium is going to take the functional units of your muscle, which are called sarcomeres, and they're going to cause them to shorten. So this is what a sarcomere looks like. It's these different fibers that are kind of resting apart from each other. And when you get the message from your sodium that just flooded into your channel, they're going to squeeze together like this, and they're going to shorten. And your muscle has repeating sarcomeres, all of them together, shorten, and that causes the muscle contraction. That was really neat, right? But you might feel a little overwhelmed. So what do you really need to know about that? I want you to know about the types of membrane proteins, sodium pumps, potassium pumps, sodium channels, potassium channels, whether or not they are uniporters, symporters, and whether or not they are antiporters, whether or not they use energy or not use energy. I want you to know the types of molecules that can diffuse and those that need to be using a protein. I want you to know the types of pumps, and I want you to know the ADP to ATP cycle. So that's dehydration, synthesis, and hydrolysis, and what a membrane potential is. To help hit these ideas home, I have 2 videos on the Blackboard for you. These 2 videos are really awesome. I like this one a lot. It shows the process of the action potential, which is really cool. And if you have any questions, email me. I hope that you have a lovely rest of your day.